I want to make this video for my patients who have been experiencing chronic enterococcus infections that you believe could be the cause of your IC and bladder pain. And I want to demonstrate that how oxalates can be playing a role in this. First, start with this journal here that I found, which is a case study that was published in 2020, which is identification of Enterococcus faecalis in a patient with urinary tract infections. Okay, so they're using next generation sequencing similar to what Microgen uses. The background basically talks about how urinary tract infection is one of the most prevalent community-acquired and hospital-acquired infections, and often caused by different microorganisms, commonly E. coli, Klebsiella, Proteus mirabilis, Enterococcus faecalis, and Staphylococcus saprophyticus. And of course, they can occur in women of childbearing age, elderly people, or people who have low immunity and urinary tract abnormalities. It says that theoretically, once the bacteria has invaded the urinary tract and contribute to urinary stone formation, it can trigger UTI easily. And of course, then this can contribute eventually to a kidney infection. So then I found this other journal. So this one was from China, and it was published in 2021. And again, just kind of some general information about urinary tract and urinary stones and UTI. And previous studies have shown that urinary stones are closely associated with gut or urine microbiota. And calcium oxalate stones are the most common type of urinary stones. However, the profile of the urinary tract microorganisms of calcium oxalate stones with UTI is not clear. So this study tried to get into what the difference was between two clusters of patients. They had two different groups, and they were looking at basically their phyla of their microbiota. And so they distinguished the two groups based on the phyla. And so we had the Firmicutes and the Enterobacteriaceae. So they found that the Enterobacteriaceae enriched in the first group was positively correlated with several infection-related pathways and negatively correlated with a few antibiotic-related pathways. But in the second group, there was more probiotics with a higher abundance, but also the good bacteria, such as the bifidobacteria, were also positively correlated with antibiotic-related pathways. And they did have some common pathogens, but it looked like, from what I'm reading, that the first group was more associated with the infection. So this kind of goes along with what I've seen with stool testing, that there is definitely an imbalance in the gut microbiota, which I'm seeing low levels of bifidobacteria and higher levels of the Firmicutes. And we also do see the Enterobacteriaceae. It just goes on to say the urinary stones are common urology diseases, and this is going on to say that most urinary stones are composed of calcium oxalate, but they could be uric acid stones, and infection stones occur in 10% of them. And then there's rare stones like cystine stones, which are 1%. And again, this is a Chinese study, so obviously, the results are from China, but the prevalence of urinary stones is 6.4% in some areas, but it can be 10% in other areas. The incidence of their formation is related to age, gender, race, dietary habits, genetics, and metabolism, which is consistent with what I've been saying. And urinary stones are closely associated with microbial taxa. So Oxalobacter is known to maintain oxalate homeostasis in the gut, which is what I've said. I said if there's certain antibiotics that we've taken during our life, we are actually depleting or destroying the population of Oxalobacter. And that the species was negatively associated with calcium oxalate stone formation, which then it continues to say that several short-chain fatty acid producing bacteria were reduced in the gut as well. So this is, again, consistent with what I'm seeing in stool testing, which a lot of my patients are also not producing. They don't have a good abundance of the short-chain fatty acid producers. And then it proceeds to say the urinary microbiome has a great impact on urinary stones than the gut microbiome. Male patients with urinary stones had different urinary microbiome compositions and significantly lower microbial diversity. Again, lower microbial diversity. And when I do the stool testing, I see this. I see lower alpha and lower beta diversity. Stones and infections always occur simultaneously. The relationship between urine microbiota and urinary stones exists. Mouse model studies show that E. coli was associated with crystal aggregation during calcium oxalate-induced renal elite lesions. So again, E. coli is associated with crystal aggregation during calcium oxalate-induced renal lesions. Protein isolated from specific E. coli strains may induce calcium oxalate crystal aggregation. Microbiota may also adhere to the stone crystals by upregulating inflammatory proteins in the urine. In this study, we focus on analyzing the urinary microbiota in these, obviously this calcium oxalate group, and found that the higher abundance of bacteria in the first group and second group were different. The Enterobacteriaceae was predominant in the first group, while the composition of highly abundant bacteria in the second cluster was more diverse. So what they're saying is the first group had more of that composition, whereas the second group had a more diversity. And this is why I keep talking about the rainbow diet and, microbiome mashup and improving the diversity, because there was an association with the gut as well. And they looked at the abundant taxa between the first and second clusters, and the urine microbiota of the second sample had greater diversity. It did have the common pathogens, but they belong to different phyla and families. In contrast, the dominant microbiota of the first cluster was this Enterobacteriaceae, 80%, which contains several pathogens such as E. coli, Klebsiella, pneumonia, and Salmonella enterica. These bacteria are often reported to be related to UTI, intestinal inflammation, and multi-drug resistance, and are the most common non-sociable pathogens. Enterobacteriaceae was closely related to bacterial invasion of epithelial cells, meaning they're more likely to cause the biofilms because they invade the cells. They also do this other pathway they're called the siderophore group. You don't need to understand all that, but this is another way that they invade your bladder lining. So the oxbacter and then the good bacteria, such as the bifido and the lactobacillus, were different in the first and the second cluster. So what they're saying is the people who were more susceptible to UTI had more of the Enterobacteriaceae, and they were depleted in bifido and lactobacillus and oxbacter. Again, why we focus on the gut, because the gut seeds the rest of the body, including the bladder. It doesn't happen overnight. It happens very slowly how this happens. So the Enterobacteriaceae and then also Proteus was also different between the two groups. And the Proteus can produce urease, which can decompose urea into ammonia. Remember, that's another thing we check in our stool testing, that ammonia content, and carbon dioxide, and produce highly alkaline urine, which is conducive to the crystallization of a magnesium ammonia phosphate. And again, these crystals can be part of the problem with some of the inflammation and the pain and the dysbiosis. Why is the Enterococcus there? Well, it is an oxalate-degrading bacteria. So an oxalate-degrading Enterococcus was isolated from human stools under anaerobic conditions, and the bacteria requires a poor nutritional environment and to maintain their oxalate-degrading ability. Calcium oxalate and its combination with calcium phosphate are major components of urinary stones. Mild hyperoxaluria is implicated as one of the causes of idiopathic calcium oxalate stone formation. The majority is derived from endogenous metabolism of glyoxylate and ascorbic acid, and 10 to 20% is derived from oral ingestion. Again, why I talk about the endogenous piece is more important than the diet, although you still have to reduce the diet because you're still contributing about 10 to 30% from eating them, but majority is derived from endogenous metabolism. And this glyoxylate pathway also occurs when there is blood sugar dysregulation, seen in diabetes, as well as in people who got this reactive hypoglycemia. The colon is the major site of oxalate absorption. 3 to 5% is absorbed under normal conditions, but the normal daily intake of oxalates range from 70 to 920, but among vegetarians, it obviously is higher. It's 80 to 2000. On low calcium diets or fasting, free bioavailable oxalate increases, and the decrease of calcium-bound oxalate and oxalate absorption is increased. Excessive absorption of oxalate may lead to hyperoxaluria and enhance the risk of urinary stone formation. The presence of oxalate-degrading bacteria in the human intestine was first reported in 1940, and they found that one main one was Oxbacter formigenes, which is an obligatory anaerobe and means that it doesn't use oxygen. That's another issue we're finding with people who are, having dysbiosis, that they have more of an increase in firmicutes and lower bacteroides, and that's often due to too much oxygen in the gut from poor digestion. So that's another thing. If there's not enough fermentation, then this bacteria cannot thrive. And among the stool specimens obtained from 5 healthy adults, They found that there were, different types of gram-positive and gram-negative bacteria. So there's not just Oxbacter, there's others that degrade. We know E. coli is one of them, but in this study, it says all 11 strains of the gram-positive were identified as Enterococcus faecalis or faecalis, however you want to pronounce it. So it seems to be when there's a lot of oxalates in the GI tract for whatever reason, whether endogenous or you're consuming it in your diet or you're absorbing it too much, you are going to essentially create an upregulation of Enterococcus. I want you to see how there's this dysbiosis link to hyperoxaluria. Hyperoxaluria leads to dysbiosis and drives selective enrichment of oxalate metabolizing bacterial species. Now, obviously, this is for recurrent kidney stone, but it can also apply to you guys, even if you've not had kidney stones. You don't have to have a kidney stone in order to be producing oxalates in your kidney or stones or crystals and passing them and causing irritation to the bladder. So just keep that in mind. These journals may be focusing on just the stones, but it can be extrapolated as well. Members of two bacterial phyla, Firmicutes and Rhabdoides, which we do on our stool testing, they dominate endogenous gut microflora, and they have complex interactions among themselves and with the host to endure stability in the ecosystem. Increased abundance of firmicutes has been linked with metabolic disorders such as obesity and diabetes, which having diabetes alone can also increase your risk of hyperoxaluria, just an FYI. Thus, augmentation of firmicutes in hyperoxaluria can be easily perceived. Bacteroides are often involved in metabolism of complex polysaccharides, mainly derived from food. So, as we discussed, and when I do your stool testing, that the bacteroides do very well on more fiber and more polyphenols in the diet. But it's been shown that this copious phyla possess contact-dependent interbacterial antagonism, which is essential to maintain stability of the gut microbiota in healthy subjects and also presents a barrier to most of the pathogens. So basically, Bacteroides acts as a natural antibiotic and keeps the levels of proteobacteria, which are more pathogenic, down. Decreases in Bacteroides are often seen with increases in proteobacteria in these particular types of subjects that they were studying. There was higher number of Prevotella as well. This indicates that above certain concentrations, oxalate could be toxic to common gut inhabitants. Similar results have been observed with respect to Oxbacter. So basically what this is saying in this, too much oxalates. So vegans or people who are consuming lots of oxalates or people who have a lot of mold in the gut, which also has an oxalate component to it, could be actually causing Oxbacter to die as well, because Oxbacter, as Susan Owen says, does not thrive in a high oxalate environment. It does better on a low to medium oxalate environment. And this is the same thing with the oxalate driving the dysbiosis is basically what this is saying. In conclusion, our study using high throughput DNA sequencing reports dysbiosis of gut microbiota communities in recurrent oxalate kidney stone sufferers. And it highlights augmentation of structural and functional diversity of oxalate metabolizing bacteria in stone formers. So the people who've got stone also have more of these oxalate metabolizing, like Enterococcus. So here's a side-by-side comparison of a test that also uses PCR. So the technology is the same here. To understand that the microbiome effects is not PCR, it is whole genome sequencing. And that's the stool test I've been using up until now, although I am starting to experiment with a new one from Genova called the GIFX, which I'm actually going to be testing on myself. But for now, that's the one I've been using. That's whole genome, different ball game. But the good news is these two technologies are the same. And I have the Enterococcus. I have a side-by-side comparison of urine on the left, which is your microgen, and on the right is this GI map. Both use PCR. And as you can see, in both cases done at the same time, this patient had an overgrowth of Enterococcus and Staphylococcus in both tests. Now, other things also did show up, like Morganella and Streptococcus, which at the time of the urine did not, but it's likely that that may have either not affected the urine or it could have been in the biofilm. But at that time that she had the bacterial load, it was definitely matching up with the GI tract, which again confirms my theory that this is a gut problem. And this is why we have to do some of the interventions that you do with me, with improving the overall composition of the GI tract, also downregulating inflammation, upregulating antioxidants. And a lot of that has to do with your diet, because your diet is going to contribute to what's growing in your gut. So again, I just wanted to make this video to show you that I do believe I'm on the right track with how I treat IC at this current moment, and why I believe antibiotics are not the solution. Thank you for listening. Bye-bye.